Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Samuel Minot. He's a staff scientist. He's part of the Microbiome Research Initiative at the Fred Hutch Cancer Center. We're going to be talking about the virome. It's a part of the microbiome, but it's the viral component of the microbiome. There's bacteria, there's viruses, there's fungi, yeast, and God knows what else in there inside of people. But there's a lot. So the virome is not often talked about. So Sam, thanks for coming.
Happy to be here. Thank you for inviting me.
Yeah, if you would tell me a bit about your research. What does it involve?
Absolutely. So I'm a microbiologist by training, and that means that, when I was going through graduate school, I started out by focusing on these different microbes that live on the human body. There's, many different types of microbes living on all of our different body surfaces, and they're really important for our health. And I started out by really looking at individual microbes and growing them in the lab in a petri dish and that sort of thing. And what the real question at hand at that time and still today is, how do the microbes on us impact our health? How is humans' health influenced by the microbes that live on us? And so this is what we think of as microbiome science. And so going from studying a single bacterium that might be able to be grown on a petri dish to the hundreds or thousands of different types of bacteria that might be found on a person and that might be influencing their health in some way.
Complicates the picture because the microbes trade resources, and one may metabolize one thing and that becomes feedstock for another, and back and forth again, and they all interact.
Absolutely. It's a complex system. Whenever we learn something new about what these microbes are doing, we find that there's these additional layers of complexity. Did we even know that bacteria could build a giant needle which they use to inject their neighbors like a harpoon and kill them when they're competing for resources? No, this is something that we only discovered in the last decade or so. There's all these types of novelty of the bacterial world that are really fascinating. And in my graduate research, I started focusing on the viruses that infect the bacteria that live on the human body. And so even one step over and above that. And actually, I did that work with Dr. Rick Bushman, who I saw had a recent podcast episode here.
Yeah, he was great to talk to. So I'm excited to talk to you too. So the viruses that infect bacteria, they call them phages, I guess, or bacteriophages. One thing I learned recently is I thought it was one virus, one phage, but I guess many different types of phages can affect one bacteria.
That's right. There are many more different types of phages than there are bacteria. And this is partially because bacteria are constantly evolving to become resistant to phage. And then those phage evolve to be able to infect those resistant bacteria. And so the back and forth, the arms race between bacteria and viruses is something which can drive the creation of new species, which is really interesting from an evolutionary standpoint.
Well, what if it's not just, predator-prey? What if the bacteria, a given bacteria's microbiome is its virome? And what if some of the viruses are helpful to it and provide tools or resources? Instead of it having to use a plasmid to talk to other bacteria or to get an antibiotic resistance gene, maybe the viruses do that for it, and then other ones prey on it. Maybe some help, some hurt. Maybe it's a whole community of things.
I think that's absolutely the right direction to be going in with this system. When we think about bacteria and viruses, we start out by thinking about predator and prey, because there's some reality there. Viruses are able to infect and kill bacteria, so bacteria are prey to those viruses. But then, as we find with all things in biology, as we learn more, we find that there's many more different varieties of that relationship. So some viruses, in fact, are found within the genome of their host and are able to emerge, be born from the bacteria without really killing them. And that's a different type of life cycle for a virus. It's still a virus, but it doesn't have that predator-prey relationship. And this is actually one of the examples that provides a really interesting connection even to human health. There was a recent discovery by the Balicki lab where they found that a bacteria that causes inflammation in the context of wounds, when you have like a cut on your skin, they found that there's this one bacteria that can cause an even worse infection of humans if it has its own virus, because the bacterial virus ends up being recognized by the human immune. And so there's many different layers always going on with any of these systems.
Yeah, I heard the example, they say it's turtles all the way down. I guess that means that, it makes you wonder. I mean, I don't know if this is possible. What if a virus itself or a phage itself had its own, microbiome? I don't know what it would be. Maybe other viruses or prions, or I guess it can't keep going forever, but it's gone several levels at least.
The one additional level that people have described is something that we call a satellite phage. And the basic idea of a virus is nothing, particularly insightful or revolutionary. A virus really is just a piece of genetic material that encodes enough information to let it spread between cells in this free-floating viral particle. And so a satellite virus is a piece of DNA that encodes enough information to put itself inside the viral coding of another virus. And so that's definitely the next area where you have these layers and layers upon them. Even something like that can be important for human health. If you think about cholera, we all know about cholera. It's an extremely important human disease. Diarrheal diseases are one of the most important targets for public health in the world today. And cholera is caused by a toxin which is encoded by a set of mobile elements, which include a satellite phage that are integrated into their host. And so there's a virus that's going around carrying the gene that causes the disease of cholera.
Huh. So cholera happens when, I guess, the Vibrio cholerae are infected by a certain phage that carries a gene that endogenizes into that bacteria and now makes it virulent to people?
That's right. The Vibrio becomes Vibrio cholera when it is infected with a phage that encodes this cholera toxin. So the cholera toxin is carried on the phage genome, and it's able to insert itself into the genome because of other accessory satellite elements that are present there. And that was work from Matt Waldor and John Michelanos at Harvard. John Michelanos is actually one of my mentors during my training. And this really, I think, gets to the interesting thing about viruses is if you just think about viruses in the world in general, you could say, well, they could do anything. There's so many of them. There's so many different bacteria. The world of possibilities is immense. And that's why I really like working at a translational research center like Fred Hutch, because at the end of the day, what we really care about is discoveries that end up having an impact on human health. And partially as a scientist, this is appealing because we only know that we really understand a system when we're able to manipulate it. And designing a good therapeutic or a good diagnostic, demonstrates that we understand the biology, but also it helps us prioritize. What do we choose to study? What do we choose to spend our time on? And when I think about the microbiome and I think about the viruses in the microbiome, I'm really interested in trying to understand which parts of those biology end up having an impact on human health. I think that's a very clarifying question.
So what are you working on right now? What's your research looking at?
So I'm very lucky in my position at Fred Hutch because I exist within this Microbiome Research Initiative. So this was a commitment by the center which said, we would really like to enhance and support and drive innovation in microbiome research because it appears that there are all these connections between the microbiome and human health. For example, with the effectiveness of certain cancer treatments seem to be related to what's in the microbiome. There are many different disorders, recovery from graft versus host disease. There's a lot of different elements of human health that might relate to the microbiome. And as a center, we'd like to do a better job. We'd like to support this area of science. And so in my work, personally, I'm a computational biologist. I spend all of my time looking at the computer. And so my work really focuses on how do we measure what's in the microbiome, given that most of it has never been cultured, has never been grown, and that we don't really know what a lot of the different organisms are doing. And so the work that I focus on is collaborating with this amazing different research laboratories that some might be infectious disease physicians, some of them might be basic researchers, some of them might be oncologists, and working with them to help understand their data that they're generating, connecting the microbiome to human health.
Well, a lot of bacteria, I guess, cannot be cultured in a petri dish, right? You can't. That's what I've heard. That make up our microbiome. Like, for instance, with that question, why do you think that is? What's stopping us from being able to culture all the bacteria?
I think there are two elements of this. One is, what is the technology that we use to grow bacteria? And I think there's a lot of different elements of that. What are the right set of nutrients? What are the right signals? For example, does this bacteria need to be in an oxygen-free environment? Can it tolerate a very small amount of oxygen? These might seem like very minor questions, but they can have a big impact on whether or not one particular bacterium actually grows. And I think that's maybe the technical answer. I think there's a larger answer here, though, which is how many bacteria are there out there in the world? How many of them could we possibly grow in the lab? And the answer is there are many. There are many, many different bacteria out there in the world. And that's not only at the level of an organism that would give a name to, for example, E. coli or Bacteroides fragilis, but also there's an amazing amount of diversity inside those species. So if you were to take one particular isolate, if you were to grow one example of Bacteroides fragilis, we grew one from me and we grew one from you, they would be different from each other. There might only be a handful of genes that would be different, but there might also be hundreds of genes that are different. And so when you find a new example of a bacterium, you find that there's always something new to discover about what that bacteria might be able to do. And I think that's the larger challenge of using culture-based methods or trying to grow bacteria as a method of studying them, as opposed to studying them without growing them, which is what I spend my time on.
Yeah, if you have E. coli in you, how many different kinds of E. coli do you have? I don't know if there's a name for it. If they have one or two genes that are different, is it a new strain? Is it not a new strain? And then, maybe it's a mistake to just say, oh, there's a lot of diversity in this microbiome. It's meaningless. Maybe we have to look at the metabolomics of it, the function of the bacteria. What are they doing in this particular context, in your colon with this mix of bacteria? How do you figure this out?
So the first question is a bit of a hard one to give a definitive answer to. How many different strains, how many different types, how many different varieties does each person have? And it can be hard to tell because the numbers are so large. If you're thinking about hundreds of millions of cells in a single person, we're not going to be able to check every one of those. But there has been some really careful work done by a couple of different groups, including, I believe, Sean Gibbons here in Seattle. And I think that the best analysis I've seen indicates that for most species, people tend to have a small number of strains. Maybe one or two strains, maybe something like 5 strains for some bacteria, but that each person has relatively limited numbers and that these are going to change over time. They might evolve some and they might also be replaced. So the strain of E. coli that I have is going to stick around in me for a while, maybe weeks, maybe months, maybe years, and at some point it might get replaced by a different strain that comes in from outside or arises through some evolutionary process, and then it sort of takes over. And that this is a constant process. There's this periods of stability and change where you have relatively small numbers of strains of each species that are each trying to maintain themselves in their ecological niche.
So what would be a better approach? Right now I know there's 16S sequencing, there's shotgun sequencing, but it doesn't seem like we're able to tie genetic sequence or genes to function. Are there any radical or different approaches people are looking at to understand what's going on inside of us or inside of some given model?
Well, this is where I have to sort of give full disclosure. I've been sort of leading us down this path because this is the area that I've been focusing on. And so I've been working on this with my collaborators in a way which I'm hoping will derive a bit more insight and understanding from the data. And I think the one way to approach this, so you mentioned metabolomics, you mentioned functional analysis. I think those are interesting areas that people are working on. Another is to try to take a close look at what we mean when we talk about strain differences. So for example, we could start the process and say, I have one strain of E. coli and you have one strain of E. coli, and they're different. Therefore, we understand nothing because they are different. The way that I tend to think about it is strains of bacteria can be more or less similar to each other. And one way in which they might be more similar to each other is in which genes they encode. And so we could go through and say, one particular strain of E. coli may contain 2,000 out of a possible 10,000 genes. Your strain has one set of 2,000 genes. My strain has a different set of 2,000 genes. If we look at what genes are found, my hope is that that will help us understand how those functions might relate to the health of the people that hold those strains. So this is gene level analysis that I've actually been spending a lot of my time on recently for microbiome research.
When you know a particular sequence, though, how do you know what that sequence does? How do you know how it's expressed?
It's pretty rare. I'd say that when you look at a sequence from the genomes of bacteria that live in the gut, you can tell what the amino acids are that are encoded. I'd say that our algorithms are pretty good at doing that. And then when you go and check it against the database and say, what other sequences have I seen like this? What other genes where I know what the function is? It's actually kind of a low rate. You might only get half of the time had any sort of name that you could give to a gene. When you do have a name for a gene, you might have a general idea of what it does. It might be a histidine kinase, but there are many different things that those enzymes could do, and it could be that there's some biological novelty there. And so I think that the annotation problem, having some idea of what the function is of the things that we're measuring, is definitely a hard one, and that's one thing that the field struggles with.
So even if you know what a sequence is, and if you know some of what that sequence does or produces, the proteins it encodes, et cetera, the enzymes, there's still a tremendous amount of variation, again, depend on context, what it could do.
And it would take us until the end of, the heat death of the universe to try to figure out the function of every gene. We could just start cataloging all the genes and saying, here's a set of 6 million genes. Let's go through and try to find the function of each one and try to clone it and express it and do the structure and do all these things. And each one would take, years of effort. And at the end of the day, we could spend our whole lives doing this, and we don't know that the genes that we've been characterizing are the ones that really matter. And I think this is where the microbiome science that's emerged over the last decade really turns the problem on its head and asks, which of these things are important for human health? Let's start with that. Let's prioritize the list of genes out there in the world and say, yes, we have no idea what this gene does, but we know that if we see this gene in someone's gut, they're at more risk for developing disease X. That gives us a good reason to say, let's start by studying this gene. That's a good one to start with, as opposed to picking randomly.
Okay, that's true. Is there a name for the translation of a given sequence to a given function? I mean, does that even have a name?
This is an area that people work on. I tend to call it functional annotation. The idea of function, I think, is something that we give names like enzyme or butyrate producing. That's sort of the language of function. And sometimes these can be things that are derived based on homology or similarity to other genes that are known. And that's something that people are working on quite a lot. There's some interesting new work coming out where people are trying to use machine learning algorithms or artificial intelligence to predict the structure of proteins that have never been crystallized before, where no one's ever found it before. And there's some developments like that I think might prove to have some really interesting developments in the functional annotation field over the next few years.
I mean, even within viruses, is it any easier to figure out what the sequence in a virus will do? I've heard, there's all these sequences we've never seen before. We have no clue what they do. There's conserved regions. There's non-conserved.
Yeah, the domain of unknown function is probably the best characterized one, or the one that we find the most often. Viruses have some hallmarks. There are some characteristics of viruses that we see again and again, and those tend to be, relatively stable landmarks. A virus has to have, or many viruses have capsids. They have proteins that coat them and protect them from the environment. They often have tail fibers. So this means they have long flexible proteins which are able to reach out like the arms of an octopus and reach out and touch and grab onto their host that they want to infect. And they also have to have some way of getting their genome into the cell that they infect. And these are usually spike proteins or tail sheath proteins. So there's some hallmarks that characterize the major families of viruses that we know about. And those can be recognized even if they are extremely distantly related to any virus that we've ever grown before. The predicted structure tends to be relatively conserved. And then on top of that, you have all these genes where we have no idea what they do. And there's a lot of room for discovery there.
So what are some insights or some important questions that you think are going to be answered in the near future because of your work or because of others' work that's ancillary to yours?
This is something I think about. This is something that I get asked a fair amount. Where are the places where microbiome science might have the biggest impact in the coming years? What are the first big things that we will see? And I think it's good to focus on this because there's an immense world of possibilities, but some of those have been carried further than others. Some of those we know a lot more about than others. One that I'm particularly interested in, partially because I work at a cancer research center, is the way that the microbiome influences the treatment of cancer. And when I was going through graduate school, maybe 10 years ago or so, if you had told me that the microbiome influences cancer, I would have been extremely skeptical. And my mind started to be changed about 4 years ago or so, as I started to see papers starting to be published where it seemed that there was some effect of what microbes are in the gut to whether or not a particular immune-based drug would be able to treat cancer. And then it all started to come together. This connection of the immune system to the microbiome is something that I think is going to be one of the biggest areas of discovery within our lifetimes. And this is because our immune system is naturally oriented towards recognizing what's in the gut. This is a major part of human development. If we grow up without a microbiome, we do not develop the immune system that is as robust as that we have naturally. And so the immune system is always sampling and influenced by what's in the gut. And so the idea that the immune system then would be poised to respond to something like cancer or cancer treatment differently is something that is seeming increasingly plausible as we start to gather more data, we as a community.
Well, do you think the immune system comes just from the perception or point of view or abilities of our somatic cells, or do you think it's a collaborative, informed effort from our microbiome as well?
I think that it's probably best not to try to think about any sort of collaboration of microbes with humans. The evolutionary timescales are so different that I think that we can think about microbes as doing their own thing in their own self-interest, and we're also doing our own thing in our own self-interest all the time. But there is certainly a tolerance that has developed where our immune system knows that certain microbes that do certain things are perfectly safe to be tolerated. And I think it's that establishment, that signaling of immune tolerance as opposed to inflammation that ends up having a large effect. If we're just sort of to boil this down to the big concepts, when you get an infection in the gut, your immune system has to change from saying, yes, there are all these microbes in my gut and I'm going to tolerate them. It has to switch its program and say, oh no, now there is a dangerous microbe in my gut. Like if you get food poisoning, you get, Shigella or something like that, then it has to change the way it responds in this physical environment. And I think that that's something that is always going on, this constant interaction between the immune system and the microbiome.
Do you think that, the virome and the microbiome, are they, I guess the virome are major players, or perhaps not, they're more major players with the bacterial part of our microbiome or with our human cells? Same thing with fungi and other creatures that are in there?
Yeah, I think that as we start to learn more, it's good to shy away from these claims of knowledge when we're actually maybe just haven't found it yet. If you ask me if a bacterial virus would influence the infection of a human, before 2 years ago, I would have said, oh no, probably not. I don't think you're really going to find that. If the virus, if the phage itself is changing the way a bacteria is able to cause disease, And then we found this example, or the Balicki lab found this example in Pseudomonas of a Pseudomonas bacteriophage recognized by the human immune system, which then makes the Pseudomonas able to infect more readily. And until that happened, I never really would have guessed that it would have been possible, but it does seem to be possible. And so we need to be ready with an open mind to change our expectations about what the world discovery will bring.
But if you're supposed to have an open mind, why would you shy away from, I guess, what some people would say, anthropomorphic language? It's, if we keep saying, no, it's just all mechanical and, nothing has thought or action or anything, and then we keep finding that there's more than we suspected, why not take the opposite perspective? What's wrong with that?
So when we try to open our mind, one way is to open our mind to sort of the way that the human brain naturally works, where we look for patterns, we try to superimpose the patterns we've learned other places in life. The pattern of how humans behave is a useful one, and we understand that as human. And so we can superimpose that on bacteria or on viruses and say, what if this virus were acting like a human? What would it do then? And sometimes we can discover something, but we have this other approach for having an open mind now, which is following the data. Instead of starting out with a clear presupposition of how this system works, we can start out by just letting the data tell us. And that's really what this amazing world of big data analysis that we live in now lets us do. Instead of saying, I think that there are 20 different pathogens, I'm going to look for those 20 pathogens in the gut and find which one of those are associated with cancer. That is presupposing that there are only 20 different things that are known to be bad, the pathobiont idea. Instead, you could just collect all the data and say, I'm going to measure all the microbes. I'm going to find which ones are associated with disease. And I'm going to let the data tell me what the biological mechanism might be. To me, that is the most exciting way to do microbiome research right now.
Okay, that makes sense. Well, very good. What do you think is in store for the next few years, 2, 3, 5 years in terms of your research and the microbiome in general? Any breakthroughs that you sense are close?
Well, I'm rather encouraged by some of the, not my own research, but I'll just say that some of the work that's bringing microbiome-based therapeutics closer to the clinic is something that I find to be extremely exciting. I think that we know a lot more about the microbiome now than we did 10 years ago or 20 years ago. And at some point, we have to be able to, sort of demonstrate this as a field. So there's one sort of biotech company I can think of that's moving forward with developing cocktails of bacteria which are observed to have an effect on human health. So treatment of, for example, C. diff infection, or even for this immune checkpoint inhibitor, immunotherapeutics for cancer. The idea of having a microbiome-based therapeutic is something that I think would really be transformational for the field and also have a meaningful impact on human health. And I think that there are a couple of leaders that are showing that this could be possible. And I'm very excited at the possibility of a place like Fred Hutch Cancer Research Center existing at the interface between basic research and translational research and cancer care. I'm very excited at the possibility of the findings that are coming from this, really exciting group of scientists being turned into something that is an actual therapeutic used in the clinic.
Well, very good. Sam, what's the best way for people to find out more about your work at Fred Hutch and to start learning about what's happening in terms of human health and the microbiome?
Absolutely. We have a page for the Microbiome Research Initiative at Fred Hutch, and I think there are some good links there. I also maintain a blog personally. So my last name, minot.bio, B-I-O. I regret that's not been updated terribly much recently, but I hope to update it more in the future. Hopefully there's more things I can also pass along.
Okay. Well, very good. Well, Sam, thanks for coming on. It's been a good talk, and I always feel like the people I talk to have so much work to do. It's insane. So I feel like that as well. There's way too much to figure out. So good luck, and I'm glad you're on the path.
Thank you. It's a busy time, but it's always good to take a minute and get to talk about science with the larger world. So I appreciate you giving me the opportunity.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.